Alrighty. Hi, everyone. Welcome. I'm Scotty Sims, the Executive Director of KCNQ2 Cure Alliance, and I'm really glad you're all here today. We know your time is valuable, and we know that joining a webinar often feels like, you know, that you're here because you care about your child and your family and, what options there are to help us in the future. Our goal today is to make this time helpful, clear, and supportive as possible. Today's webinar is an introduction to Praxis's Emerald study for KCNQ2 individuals. And this session is focused on sharing information about the Emerald study, a clinical research study led by Praxis Medicines. And this study is evaluating an investigational medication called relutrigine, which is being studied as a potential treatment to help reduce seizures associated with developmental and epileptic encephalopathies, for short, DEEs. So we're grateful to be partnering with Praxis Precision Medicine's team today. So joining us on our call is Julia Chase. Julia is the Senior Patient Engagement and Recruitment Specialist and Patient Advocacy Lead. And Julia's been with Praxis for 5 years, and her work is primarily focused on the DEE portfolio. And today's presentation will be led by Kelly DeReal. Kelly has worked at Praxis for 7 years, and she plays a key role in driving the DEE studies forward. Her work is deeply personal and is driven by passion. 10 years ago, her son was diagnosed with a genetic epilepsy linked to the SCN2A gene. And this experience inspires her to dedicate her career to advancing treatments for seizure disorders, not only for her son, but other children and families facing similar challenges. Today, she'll walk us through how the Emerald study is designed, who may be eligible, and what participation would look like for families. We've set some time aside for questions at the end because we know families often come with very real and very specific concerns. So a few housekeeping rules we have before we begin, just so everyone feels comfortable participating. Make sure you keep all your microphones muted. And if you haven't already, please open the chat box so you can ask questions either by typing them in the chat or using the raise the hand feature once Kelly's presentation is over. We're recording this presentation today, and the recording will be shared afterwards. So if you want to revisit with any questions. With that, I'm happy to turn things over to Kelly, who will walk us through the Emerald study. Kelly, Julia, thanks for being here.
Thank you for having us. All right, I'll pull up some slides. Kelly, just let me know once they're in presentation mode.
Yes, you can flip to the next slide. All right, thanks. As Scotty mentioned, my name's Kelly. I'm the Director of Patient Strategy at Praxis, and I got into industry about 11 years ago when my son Connor was diagnosed with an SCN2A mutation and really wanting to work on new and better treatments for DEEs. I'm not the only person that works at Praxis that has a child with a genetic epilepsy. There are several of us, which means the patient voice is coming into our meetings on a daily basis. In Praxis, really, our DEE studies, we're not just following the traditional clinical trial designs. We're really about breaking the status quo and how do we make trials more patient-centric. And one way we're doing that, and what we'll get into in a later slide, but we're bringing the clinical trial to the family's home and removing the burden of travel altogether so that more families and patients can be included in trials and trying new potential medicines. And then this is our pipeline at Praxis. We are an epilepsy company, so we have two different platforms. The first is a small molecule platform, and this is where relutrigine sits at Praxis, and this is the drug that we're going to be talking about today. We also have an antisense oligonucleotide, or ASO, platform at Praxis, and these are gene-specific ASO treatments. And I know for me, I trialed so many different anti-seizure medications with my son when his seizures started. And after the third or fourth, I really lost a lot of hope that the next one would be any better or any different than the previous ones we've trialed. I want to make it really clear that the mechanism of action of relutrigine is very different than what's currently available today. It's in the class of sodium channel blockers, but relutrigine is a sodium channel modulator. And what that means is it's selective to the hyperactive neuron. Most sodium channel blockers are just blocking all sodium channels. And relutrigine is relutrigine is selective to the hyperactive neuron, meaning when neurons are functioning normally, it leaves them untouched. Relutrigine steps in when there's too much neuronal activity. So it's very different. There's no titration whatsoever. Typically, with a lot of anti-seizure medications, we have to slowly titrate up over a series of weeks or even months to get to a therapeutic level to then assess if it's going to be efficacious. With relutrigine, the dose you start at is the dose that you stay at. So as a caregiver, you're going to be able to tell very early on if there's efficacy with relutrigine. The half-life is very long, about 5 days, which means it only has to be administered once a day. And the half-life is so long that if a caregiver accidentally forgets to give the medication that day, it is not going to affect the levels. For this trial, relutrigine is a liquid formulation. It can be taken orally or by G or J tube, and there's no added sugars. So for patients on the keto diet, there's no issue. And then I want to touch on the different phases or the different trials that relutrigine has been in. We started at a phase 1 trial, and this was a trial with healthy volunteers. It was tested in 133 healthy volunteers. And based on the data that came out of that phase 1 study, we were authorized to then move into a phase 2 study with a patient population. And we first trialed it in a cohort of SCN2A and SCN8A patients called the EMBOLD study. Based on an interim data cut we did on that first cohort, we started a second cohort of these patients with a lot higher numbers. And then in parallel, we just started the EMERALD trial, which is a phase 3 study. This will be the last trial that relutrigine is in before it moves to what will hopefully become a commercialized drug. And there is a pretty large gap from when a phase 3 trial ends to when it can be prescribed by a physician. A few years, best case scenario. So anyone that is interested in trialing relutrigine, this phase 3 trial called EMERALD, it would really be the last opportunity for a few years to trial this. And then I want to touch on the data that came out of the EMBOLD trial. So our very first cohort, it was a small number of SCN2A and 8A patients. And when we did the first interim analysis, we looked at all patients that had gotten through the double blind, so the first 4 months. These patients could remain on their current anti-seizure medication regimen, and then relutrigine was added on top of that. So we saw a 46% seizure reduction in month 4. The second cohort with a larger volume of patients, when we did a recent interim analysis, we saw a 53% reduction in seizures at month 4. And when we look at the 11th month cut, so patients that have been on relutrigine for a substantial amount of time that were in open label extension, we're seeing a 90% reduction in seizures. And really, this is sort of the opposite of what we see a lot of times when we're trialing new anti-seizure medications that a lot of people refer to as a honeymoon period, where we see a lot of efficacy in the beginning, and for whatever reason, that wears off and the drug is not working as well as it was in the beginning. With relutrigine, the longer that you're on this molecule, the better your seizure control seems to be. And then in addition to the primary endpoint of a reduction in seizures, we also are using several scales in EMBOLD and in EMERALD. One of those scales is called the CGI, and we pulled out 4 specific domains. So we looked at disruptive behavior, and this is whatever that disruptive behavior is unique to that specific patient. We looked at alertness, communication, and seizure severity and intensity. So how often is rescue medication being used? How long are the seizures lasting? And on the day of screening, this scale is utilized by the caregiver and by the principal investigator, the clinician. And then these same domains are reassessed at the end of the double blind, so 4 months later. And as you can see, there are significant improvements according to both the caregiver and the clinician in all of these domains. And typically, when we use sodium channel blockers, that's where children can have side effects like sedation or tiredness or not as alert. With relutrigine, we're seeing the opposite, where awareness is improving, alertness is improving. So because we're not blocking all sodium channels, we're not seeing those same side effects that we get from sodium channel blockers. And then just to touch on the adverse events that we're seeing in cohort 2 of the larger study with SEN2A and 8A patients. As you can see, there were no severe adverse events due to drug. And in the placebo group, those side effects were experienced at a higher rate. And then we received breakthrough therapy designation for our SEN2A and SEN8A patients based on the results that came out of the EMBOLD study. And this is a designation that the FDA gives to a certain compound when the other available treatments, that a new compound has better efficacy and a better safety profile than anything else currently available. And we're hoping to seek that same designation for our DEE patients in the EMERALD study, which means everything is fast-tracked and sped up to get this drug to as many patients as possible in a quicker amount of time. And then relutrigine has much broader applicability than just sodium channelopathies. So regardless of the root cause of your seizures, whether it's a KCNQ2 mutation or CDKL5, sodium channels play a role in the formation of seizures. The last step before a clinical seizure occurring, neurons have to pass through sodium channels, and this is precisely where relutrigine acts. So neurons that are firing too fast or too often, relutrigine is selective to those neurons, bringing them back to a normal state, and the hypothesis is then a seizure is not going to occur. We trialed relutrigine in a pretty large library of animal models, and I want to specifically call out one of those animal models that relutrigine was trialed in was a KCNQ2 mouse model. And we saw tremendous efficacy in this animal model with a cessation of seizures. So again, this trial is for broader DEEs, but we do have positive animal model data in a KCNQ2 mouse model. And then I want to touch on the unique trial design we have for the EMERALD study. So we have options for families. If a family lives local to one of our clinical trial sites, you can go in person, in clinic for all of your study visits. But the majority of families choose the fully remote from home option. So I'm going to talk through the study design with that option in mind. And what this looks like is on the day of screening, a physician that's licensed in the patient's home state comes to the patient's home along with a nurse, and labs are drawn out of the home. All assessments are done in the home. The principal investigator joins via telehealth. And then for the next 28 days, the caregiver is just filling out a daily electronic seizure diary. After those 28 days, if the patient has a minimum of 4 seizures and all of the labs are within normal range, then the patient is randomized into the study. And for the first 16 weeks, it's randomized 1-to-1. And what that means is 50% of patients will get placebo, and the other 50% of patients will be on relutrigine. There's a study visit once per month, and all of these study visits are occurring out of the home. Drug is shipped to the home. At the end of those 16 weeks, then families can roll over into what's called the open label extension for 32 weeks. And what that means is there is no more placebo group and everyone is on relutrigine. And when you get into the open label extension, study visits become less frequent. So they're every couple months. And then So to explain further the at-home option. So you're still being transferred to a study site. The principal investigator at that study site still has oversight of the patient and monitoring relutrigine. Your child's primary neurologist is able to be as involved in this study as you want them to be, which means oftentimes some of the primary neurologists will confer with the PI multiple time points during the study. But the principal investigator still has oversight over the doctor that's coming to your home, and can be speaking frequently with the primary neurologist. And then with regard to who can participate, so anyone with a diagnosed DEE, so anyone with the KCNQ2 mutation, that would qualify as a DEE. It's a pediatric through adult trial, so 2 to 65 years of age at the time of screening. Seizure onset just has to happen by 12 years of age. And then a minimum of 4 motor seizures. And patients can remain on their anti-seizure medication regimen. There's no cap on the number of ASMs a patient can be on, but only 2 of those medications can be true sodium channel blockers. And then any family that's interested, the next step would be, we have a trial-specific website. There's a page on Emerald. There's some information that's similar to what we just reviewed today. And you would be able to schedule a call with the nurse navigator who would take you through a series of questions on eligibility. For example, what age did your child's seizure start? What medications are they taking? If the patient is eligible, there will be a warm handoff to the study site. And within about 1 to 2 days, the study coordinator from that site will be contacting you. And this is the point where you're able to connect with the principal investigator, ask all the questions that you have. You're able to have your primary neurologist connect with the principal investigator to really make sure this is a good fit for your child and this trial is a good fit for your family. So there are some families that pre-screen, they're transferred to a study site, they review the informed consent, and they schedule screening right away. There are other families that have some interest, and it takes them a little bit of time where they want to really think this through, have their primary neurologist connect with the principal investigator, and have a lot of questions and take this a little bit slower until they make up their mind that yes, they do want to screen for this study, or maybe they come to the conclusion that it's not a great fit. So I really urge anyone that has some interest, you would want to take this next step, ask more questions, learn more about the mechanism of action, or anything else that's on your mind, and you're able to connect with the physician at the trial site. And I think we're going to open it up to questions now. And really, this is